Good day, everyone. I will be talking about gram-negative rods in the respiratory tract. So, generally, they are considered gram-negative because during the gram-staining reaction, they retain the secondary dye or the counterstain, which is your safranin, and during the decolorization process, the primary dye has been lost, thus appearing red after the entire gram-staining procedure. And upon examination under the microscope, they are rods, no? Or bacilli in morphology, although they are actually considered pleomorphic in nature because they may not look like rods all the time. Okay, varying they actually have varying morphology depending on the media that was used for the isolation and the culture condition. And these gram-negative rods reside in the respiratory tract, no? Therefore causing infections in the respiratory system. So we have three medically important gram-negative rods causing infection in the respiratory tract. We have your Hemophilus, Bordetella, and Legionella. Under your Hemophilus, the most common, the model organism is your Hemophilus influenzae, which when it infects an individual, causes sequelae, no, sequelae of pneumonia, no, a lot of consequences. However, this infection caused by this microorganism is actually preventable, uh, especially when we do vaccination. And the most common population that has been affected by this organism are your infants. In fact, Hemophilus influenzae, aside from pneumonia, it may actually cause a lot, no, a lot broad spectrum of diseases, especially the capsulated type, no, could cause otitis, meningitis, and among others. Then we have your Bordetella, uh, the common organism infecting man is actually your Bordetella pertussis. Although, aside from Bordetella pertussis, we also have other species, no, which may cause infection. So we have your Bordetella parapertussis and Bordetella bronchoseptica. We'll discuss that in the next few slides. And then we have your Legionella. Legionella, the most common species causing infection to man is your Legionella pneumophili, which causes pneumonia. So under Haemophilus species, generally they are gram-negative, and as I have mentioned a while ago, they do not just look rods or they do not just appear bacilli solely. After gram staining procedure, they are actually pleomorphic. Pleomorphic because they may appear rods, they may appear cocoid bacilli, cocobacilli, or long rods, no, depending on the culture conditions that was used during its isolation, thus pleomorphic. And these microorganisms are actually not cultured easily because it requires enriched media, special media, usually containing blood or its derivatives. Now, it's the term used here is enriched because you actually have to put some nutrients, no, some nutrient requirements that are needed for the growth of the bacteria, favoring its isolation and growth. Now, for Haemophilus species, there are actually two nutrient requirements which are used for its isolation. In other words, without these nutritional requirements, so absence of these requirements in the media would actually not let you isolate Haemophilus. No? And these requirements are the following. So we have your X factor and your V factor. So your X factor or your hemin factor, Hemin, hemin factor, is actually derived from your hemoglobin found in the blood. Okay? And your V factor or your NAD, no? NAD or your nicotinamide, okay, diadenosine. Um, this should also be present, no? For the isolation of your Haemophilus influenzae. Okay, so shown in the table here are the different important Haemophilus species causing infection to man. And to note, you actually need, okay, the X and the V factor for its isolation, particularly for H influenzae. However, for some other species under your Haemophilus, you only need either one of these, no? Except for H influenzae and H hemolyticus. Now, aside from the requirement of your X and the V factor, there's also a distinct characteristic of particular species under the genus Hemophilus, such that your Hemophilus hemolyticus, no, from the term itself, from the word itself, hemolyticus, and your para-hemolyticus, these are actually the species which are hemolytic. No? In other words, when cultured in the enriched media, and since these enriched media contain blood, causes lysis of blood. No? And so they cause lysis of blood cells in the culture media. That means these two here, okay, your hemolyticus and parahemolyticus contain hemolysin, causing lysis of red blood cells in the enriched media. Now, to note, your the three of these, no, your Haemophilus influenzae, parainfluenzae, and Haemophilus ducreyi are actually the three most important Haemophilus species causing infection to man, such that your influenzae, parainfluenzae causes the typical respiratory symptoms or infection, whereas your Haemophilus ducreyi is actually a sexually transmitted um, disorder, no, causing this what we call chancroid. Then we have your, you might ask, Doc, how come there's A here, this Agrigobacter here? So your Agrigobacter aprophilus was actually formerly called Hemophilus aprophilus and Hemophilus paraaprophilus. But now it was renamed, no, to Agrigobacter. And then we have your Agrigobacter segnis, was actually formerly known as your Hemophilus segnis, but it's later renamed as Agrigobacter segnis. So these are actually normal microbiota of your mucous membranes and only occasionally cause human disease. So let's start with your Haemophilus influenzae. So characteristically, I mentioned it's pleomorphic. Pleomorphic because the appearance, the typical morphology that is gram-negative rods is not appreciated all the time, no? Because at cultures, no, the appearance, the pleomorphic appearance would actually vary depending on the culture conditions that was used to isolate the microorganism, such that normally it appears coccoid bacilli, and sometimes it may appears pairs, no, in pairs or in short chains. And you get to appreciate the coccoid bacilli, the coccobacilli morphology when you incubate, when the cultures are incubated from 6 to 8 hours. And at a later period, okay, at a later period, the forms actually vary, no? Most probably occurring in chains, okay, or bacilli. And yeah, as I've mentioned, the morphology depends on the length of incubation. Again, if the culture was incubated from 6 to 8 hours, it most likely appears coccoid bacilli. And at a later period, okay, the morphological appearance of pairs or short chains are favored. And young cultures, isolated from 6 to 8 hours express a capsule. Okay, so you have to take note that there's actually a strain of Hemophilus influenzae which are which do not contain capsule. So we have encapsulated and unencapsulated strains, okay? And depending on the presence or absence of capsule, the type of infection that it causes actually varies. So we'll discuss that in the next few slides. So on culture, they appear flat, grayish, and translucent with colonies appearing after 24 hours of incubation. So notice the the flat, no, grayish appearance, translucent colonies on chocolate agar. However, as I've mentioned, they do not normally grow in typical or routine culture media because there is a need for you to supplement your nutritional requirements of X and the V factor or the hemin and the NAD, no? But in cases wherein, okay, in cases wherein you do streaks, okay, of Hemophilus culture alongside, no, or perpendicular to the culture streaks of Staphylococci species in normal, no, just a plain blood agar, okay, it will actually grow. Doc, why will it grow, Doc? Remember you mentioned, Doc, that the normal blood agar do not contain the needed nutritional requirements. Well, it contains the X factor because remember, the X factor or hemin are actually found on the red blood cells. And however, the V factor, although not found in the typical or the plain blood agar, the V factor is actually supplied by the Staphylococci species. Thus, when streaked, when cultures of your Haemophilus influenzae are streaked alongside or perpendicular to culture of your Staphylococci in a sheep blood agar, your V factor is actually supplied by the Staphylococci, and so it favors growth now of your Haemophilus influenzae. And we call that growth, no? We call that growth perpendicular to streaks of Staphylococci species as satellite phenomenon. So remember again, the Staphylococci actually promotes production of your NAD or your V factor, thus favoring growth of your Haemophilus influenzae. So, as I've mentioned, your Haemophilus, particularly your Haemophilus influenzae, causes disease to man. And this disease causation is actually due to the presence of several, okay, several enzymes and structure in the microorganism which promotes infection. So we have this what we call, no, the most predominant, the most common. I'd like to put emphasis on that. No, predominant and most common virulence factor of your Haemophilus influenzae is the capsular polysaccharide. But do not be misled, no, because remember I told you that there are actually strains of Haemophilus influenzae which are unencapsulated, meaning they do not contain any capsule. Although they still, okay, take note, they still cause infection. But the most predominant, most of these infections caused by your Haemophilus influenzae are those strains containing your capsular polysaccharide. And to note, okay, different from other capsules that we have encountered so far, from the gram-positive and other capsules that we have mentioned, the capsular polysaccharide of your Haemophilus influenzae is actually made up of PRP or your polyribosyl ribitol phosphate, no? A ribose, an alcohol ribitol, and a phosphate group. And to note, no, you've learned this before, that the main role of your capsule is that it acts as a shield, no? It acts as a shield protecting your microorganism from the actions of your phagocytes. Thus, it's anti-phagocytic and it helps the bacteria resist phagocytosis. And another um, virulence factor are your lipopolysaccharides. So remember, your LPS or your lipopolysaccharides are actually situated in the outer membrane. Okay, the outer membrane. Thus, it actually may serve as an endotoxin, causing inflammation, not triggering inflammation. Another is that it's capable of producing an enzyme we call IgA1 protease. So it facilitates breakdown of IgA, facilitating colonization of Haemophilus influenzae on mucosal surface. Take note that you've learned before in immunology that your mucosal surfaces are lined by immunoglobulin. Okay? And the most predominant type of immunoglobulin lining your mucosal surfaces are your IgA. And because your Haemophilus influenzae contains a protease capable of breaking down IgA, so thus the IgA1 protease, it's actually easy for it to gain entry on these mucosal surfaces. So remember, your respiratory tract are lined by mucosal surfaces. Thus, your Hemophilus actually causes easy infection in the mucosal surfaces because of the presence of these enzymes, no? Causing breakdown of IgA, facilitating colonization, and helping its adherence in the mucosal surface epithelial cells is your pili. Okay? So the disease process caused by your Hemophilus is actually very complex. No, why complex? Because it actually involves damage. You know, a lot of coordinating function of those virulence factors I have mentioned, causing damage to the respiratory mucosa. So, being an inhabitant, no, preferring to inhabit the respiratory tract of the host, okay, the portal of entry, the route of infection is actually via respiratory route, no, through inhalation of respiratory droplets. Take note of that. And upon inhalation, through respiration, upon inhalation of those respiratory droplets, chances are, if those droplets contain Haemophilus influenzae, it's actually easy for them to adhere to the mucosal surfaces in lining the respiratory tract because, as I've mentioned, they contain pili or pilus. No? So they adhere. And as someone having that immune system, no, the primary defense by your immune system in these mucosal surfaces are your IgA lining found in the surface, no, in the mucosa. However, since your Haemophilus contains protease enzyme, the protease enzyme is actually capable of breaking down the IgA lining the mucosal surface, and so it's easy for it to gain entry into the respiratory mucosa, causing damage. No, the damage is actually um, triggered by these virulence factors that I have mentioned. Okay, and so infection starts, and with high burden of bacteria of infection, the microorganism may also invade the bloodstream. Okay. However, if you are immune to the infection, okay, if you are immune to the infection, supposing you have antibodies, you have been infected before, or you've received antibodies from passive transfer, or you've received vaccination, okay, and some of your complement components, and also your phagocytes, which are basically comprising your immune system, part of the defense mechanism by your immune system, would actually clear out the infection, no, or the microorganism in the circulation. However, if you do not have these, no, you have impaired functioning of your complement system or your phagocytes, or if you do not have any antibodies against the virulence factors, particularly the anti-capsule, okay, antibody, or your antibody specifically against the polyribosyl ribitol phosphate, okay, so you will get the infection. And when the burden of infection is actually very high, that is, there's actually high replication of your bacteria, it actually causes disseminated infection. Disseminated because it's not going to affect just the respiratory tract, but also the bloodstream, the meninges, subcutaneous, tissue joints. In other words, my point here is that when there's heavy infection and your immune system could not actually contain that infection, it causes the bacteria to disseminate to other parts of the body. Okay? Causing a lot of infection mentioned here. Okay? And however, those infection caused by the non or unencapsulated strains cause infection in the sinuses, causing sinusitis and otitis. So this is actually a diagram showing us the common infections caused by these, no, Hemophilus influenzae strains, depending if it contains a capsule or not. So as I've mentioned before, that those who do not contain capsule prefer, no, they actually prefer residing in the respiratory tract, causing otitis, conjunctivitis, and bronchitis. Although conjunctivitis more of outside the respiratory tract, but still mucosa, no, lined by mucosa. However, the encapsulated type are the strains um, causing predominantly systemic infection. Systemic because it's actually disseminating or disseminated to other parts of the body or other organs, preferably in the bloodstream or some other organs such as your joints, your meninges, your heart, and among others. So these are actually triggered by the virulent strains. Virulent because this contains those virulent factors that I have mentioned. They are capable of invasion and they also release endotoxins. So the only way to prevent primary infection is through vaccination. And you receive this vaccination when you were young, no? Given between 2 to 18 months of age. The vaccine actually contains the type B capsular polysaccharide or your PRP polysaccharide, similar to diphtheria toxoid. That means um, the type B capsular polysaccharide injected during vaccination or immunization would actually trigger antibody production against your PRP. Thus, the anti-PRP antibody production. Okay. Then we have another species, your Haemophilus ducreyi. So your Haemophilus ducreyi, as I've mentioned, is also significant because it causes a sexually transmitted infection. We call that your chancroid, no, or soft chancre. It's actually a small gram-negative rods, okay, gram-negative rods, and as I have mentioned, no, these Haemophilus species actually have a very strict requirement such that it only grows in an enriched media. So for Haemophilus ducreyi, um, the requirement is that the media, okay, the media should contain, as I've mentioned, no, it should contain your X and the V factor, and it should also contain your vancomycin. The vancomycin here would actually inhibit other bacteria, no, aside from your Haemophilus species. And The agar is usually incubated in 10% CO2 at 33 degrees centigrade. The samples that is used to grow Hemophilus influenzae are the scrapings from the chancre, okay, of an infected individual, particularly in the base of the ulcers. So the disease caused by Haemophilus ducreyi is, as I've mentioned, is actually Chancroid, which is characterized by the presence of a ragged ulcer on the genitalia. So this is the ulcer typical of Chancroid, caused by your Haemophilus ducreyi. Notice that the edges are actually irregular and ragged. It doesn't have a defined edges. It's irregular. And to note, these ulcers are actually very painful, okay, to differentiate it from the ulcers caused by your Treponema pallidum infection or your syphilis, because the ulcers caused by your Treponema pallidum in syphilis are actually painless. Although it's kind of difficult to really determine this ulcer was caused by Haemophilus ducreyi, this ulcer was caused by Treponema pallidum in syphilis, but the thing is, it's the ulcers, um, the ulcers caused by the Hemophilus ducreyi in Chancroid are actually painful. Okay? And there's actually no associated permanent immunity after you got infected with your Hemophilus ducreyi. So it's sexually transmitted. Take note of that. So another medically significant gram-negative rod infecting the respiratory tract is your Bordetella. Okay? And we actually have a lot of species under Bordetella, but the important ones causing infection are the three. So we have your Bordetella pertussis, Bordetella parapertussis, and Bordetella bronchiseptica. So very common. You've heard this before. Your Bordetella pertussis is actually the causative agent of your whooping cough or your pertussis. And your Bordetella parapertussis causes pertussis-like infection, eventually leading to pneumonia and some other complications. Then we have your Bordetella bronchiseptica, which causes infections in animals, particularly pigs. So this is a table outlining the different types of your Bordetella, okay, the species under the Bordetella genus, and the different infections it causes. So for examination purposes, you focus on those the classical species causing infection that are medically important to humans. So we have your bronchiseptica, okay, as I've mentioned, causing infection in animals, particularly pigs. And then your parapertussis, a whooping cough or pertussis-like infection in humans. And of course, your Bordetella pertussis causing pertussis or your whooping. So, since these three are actually medically important, okay, the pertussis, parapertussis, and bronchiseptica, there's actually a need to differentiate the three on the basis of their growth characteristics and biochemical tests, such that your Bordetella bronchiseptica actually grows on McConkey agar. That means this is a lactose fermenter. Whereas your Bordetella pertussis is only the one who doesn't grow on McConkey and sheep bloods agar, because you actually need entirety, the all Bordetella actually should be grown in an enriched media, your Bordet Gengo medium. And to note, your Bordetella pertussis is actually a slow grower in the Bordet Gengo medium, taking it 3 to 6 days to grow. whereas your bronchoseptica actually grows after 24 hours of incubation. The only motile genus of the three medically significant Bordetella species is your Bordetella bronchoseptica, and the only oxidase negative, no, not producing the oxidase enzyme, is still your Bordetella bronchoseptica. So it also has the Bordetella species in general may also be tested for urease production. Okay, urease production meaning it produces an enzyme urease. Urease, okay, which is typical of your Bordetella parapertussis and Bordetella bronchoseptica, and capable of converting nitrate to nitrite is your Bordetella bronchoseptica, and using citrate, no? Using citrate as a source of energy is your Bordetella bronchoseptica, and sometimes your Bordetella parapertussis. Take note of this. So we'll discuss these biochemical tests in detail, okay? When we discuss your Enterobacteriaceae next week. So recently we had reports, no, local reports in the Philippines of an outbreak, no, pertussis outbreak. Particularly, there are 5 regions having reported the most number of cases, no, in Mimaropa, in NCR, in Luzon, in Visayas particularly in Dumaguete, no, in somewhere in Negros, and in Western Visayas. Okay, and you learned before that pertussis is actually caused by your Bordetella pertussis. It's actually a small, no, really small gram-negative coccobacilli resembling your H. influenzae. So it's a very small and kind of very difficult for it to visualize under gram staining, such that this is best identified using immunofluorescence. So immunofluorescence staining. So it's non-motile, and usually it's capsulated. And as I've mentioned, we need an enriched culture media. Okay, enriched because this contains additional nutritional requirements needed for growth. Okay, and as I've mentioned, Bordet Gengu medium is actually the medium of choice for the isolation of your Bordetella pertussis. It contains potato blood glycerol, and it also contains penicillin. The penicillin here would actually inhibit other bacteria which are not Bordetella, making this media or that bingo really specific for the isolation of Bordetella. That means only Bordetella species grows in Bordet bingo medium. So sometimes a more preferred, no, a more preferred media for isolation of your Bordetella pertussis aside for Bordet bingo is your Regan low medium. Okay, your Regan low medium contains charcoal, which acts as a detoxifying agent. It's also supplemented with horse blood, your cephalexin and amphotericin B. Your cephalexin is actually an antibacterial agent which inhibits growth of other bacteria aside from the Bordetella species. And your amphotericin B, which is actually an antifungal agent which inhibits growth of fungi. Thus, this is actually more preferred, no, for the isolation of your Bordetella because it contains these inhibitors. So on culture, the colonies appear as ray shiny mercury or dewdrop appearance. So look at the mercury drop appearance of the colonies. It's the shiny here, the shiny appearance here is actually imparted by the capsule, okay, capsule of the organism. So it's a strict aerobe, no? Strict aerobe because the strict or obligate aerobe because it requires, okay, it requires an aerobic environment. No? It requires an space aerobic, okay? Requires aerobic environment for growth. And aside from being an obligate aerobe, it's also capable of producing oxidase and catalase enzyme. However, it's nitrate, citrate, and urea negative, your Bordetella pertussis. Okay? So it does not require your X and your V factors compared to your Hemophilus, which requires these two factors as requirements for growth. So there are several factors, virulence factors, which are found as part of its structure causing disease, no? Which favors disease causation in man. So the toxin produced by your pertussis actually is capable of fixation. And then we also have your heme agglutinin. Okay, your heme agglutinin is actually the one responsible for adherence to epithelial cells. Your lipopolysaccharide is an endotoxin. Your pertactin, also for adherence. Your dermonecrotic toxin causes actually necrosis in vascular structures. And then we have your adenylate cyclase toxin, which um, favors, it actually inhibits no chemotaxis by your leukocytes, no, and others leukocyte function. And then your tracheal cytotoxin, which actually causes ciliostasis. Ciliostasis meaning your cilia lining the trachea actually is not functioning because of this cytotoxin, the inhibition of ciliary function, causing now impaired, no, um, impaired release, no, of these bacteria once it starts invading the respiratory tract. So, as I've mentioned, your Bordetella pertussis causes pertussis, or this is also known as whooping cough or tospirina. Okay, it's actually called whooping cough because the pattern, there's actually a typical pattern of the hacking coughs. No, it's actually paroxysmal, and um the whoop, no, in the whooping cough is actually produced at the end at the end at the end of the entire coughing cycle. It actually ends with an inspiratory whoop. Okay, so you search for the sound of the whoop, huh? As you make that as your assignment. So I'd like to direct your attention on the diagram here on the right. Okay, this will actually outline the entire spectrum of the disease or the different stages of the disease, characteristic of your pertussis, of your whooping cough. So generally, we have three major stages: your catarrhal stage, your paroxysmal stage, and your convalescent stage. Now, upon exposure to the bacteria, of course, no, you are asymptomatic because it's gonna take some time for it to cause infection. Meaning, the incubation period is actually the time period from exposure of the bacteria, you getting the infection up until the symptoms manifesting, no, in the catarrhal stage. So, in the incubation period, it's gonna take 7 to 10 days. This is actually the period wherein the bacteria is replicating. And you see here, okay, there's actually linear or logarithmic growth of bacteria, such that when you have high burden now of bacterial count, you actually cause now, no, infection. You, you cause now infection, and when infection happens, symptoms starts to occur. Okay, such that the first stage now of your pertussis infection is term catarrhal stage, characterized by rhinorrhea, no? And mild cough, no? Rhinorrhea, runny nose or coryza, and mild cough. To take note, this is actually the period wherein there's actually maximal bacterial count. And since there's actually high levels of bacteria here, the best method to detect infection is through PCR determination and culture. No, you can actually isolate bacteria here because you have high burden, no, high number of bacteria because it's replicating at its maximum. And there's also chances of getting positive PCR results here because remember, this is also dependent on the amount of bacterial load. Okay? Therefore, if your catarrhal stage is actually characterized by high levels, no, of bacterial load, therefore, this is also the stage wherein your antibiotics are most effective. Why most effective? Because this is the stage containing the highest bacterial load. And so you have the bacteria there alive, okay, causing the infection. So you have the antibiotic to kill that bacteria, okay, in the catarrhal stage. Approximately that's 1 to 2 weeks. And then we have your paroxysmal stage. No, this is actually the stage characterized by the whoop, no? The inspiratory whoop. Okay, characterized by cough and the inspiratory whoop, and also lymphocytosis and vomiting. Lymphocytosis again means there's increased lymphocytes in the blood circulation, or lymphocytosis. Then there's also characteristic vomiting. Now to note, your bacterial load starts to decline in the paroxysmal stage, and remember your Bordetella pertussis actually contains a lot of virulence factors, and these virulence factors are actually structures of the bacteria itself, no? And your immune system responds to these bacterial structures, virulence factors, through production of antibodies. So in the catarrhal stage, this is actually where there's starting to maximal, no? Beginning to maximal antibody production. Okay? Against those virulence factors that we have mentioned a while ago. And in the convalescent stage, okay, the convalescent stage, you could not actually detect here any bacteria because there's actually decline of bacteria in the paroxysmal stage. No? So PCR, your culture, are actually very ineffective in diagnosing the disease conditions, but we solely rely on serological testing. No, serological because we need to identify the antibodies against those virulence factors found in Bordetella pertussis, associated with Bordetella pertussis. No? Thus, the most common sample here is actually your blood sample, particularly your serum, no, for convalescent stage. So this is the stage, okay, stage wherein antibiotics are least preferred because remember your antibiotics kill the microorganism and you don't have microorganisms already in the convalescent stage. And also to note, your paroxysmal stage is actually the stage, no, wherein the typical symptoms or clinical symptoms of your pertussis is actually um, best seen. Take note of those. Okay, so this is another diagram from CDC. Okay, outlining the disease progression of pertussis. No? So again, we have the catarrhal stage, your paroxysmal stage, and convalescent stage. Again, which stage is where when antibiotics are its most useful, no, and its maximal effect in the catarrhal stage. So which stage is actually characterized by the typical manifestations, clinical symptoms of your pertussis in the paroxysmal stage, and which stage now, because there are no organisms and there's antibody production, so antibiotics are not effective in the convalescent. So this is another table summarizing everything that I just mentioned. Okay, so your catarrhal stage, the duration is 1 to 2 weeks, paroxysmal 1 to 6 weeks, convalescent 2 to 4 weeks, and with the following symptoms. So remember, I mentioned that in the catarrhal stage, the symptoms are actually more of viral in picture, no? Viral in picture and not typical of your pertussis, okay? As seen in the paroxysmal stage, which are very characteristic of your pertussis, no? Whooping cough, the lymphocytosis, and the vomiting. And then your convalescent stage, um, this actually may predispose an individual to other respiratory infections. And this is also the stage wherein some other complications arise. So as I've mentioned before, your polymerase chain reaction is best done within the first few stages of infection, particularly once the 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 most high yield stage during your when doing your PCR and culture in the catarrhal stage. Okay, because that is the stage wherein you have the highest bacterial load. And serology or serologic testing is best done during the convalescent stage because in serology, you only detect the presence of antibodies. No more detection of the organism. And as I've mentioned, the same with Hemophilus, this is also preventable, okay, through vaccination via the Tdap vaccine, no? Diphtheria, tetanus, and pertussis vaccine. So this is given from 6 weeks to 6 years of age, and some booster dose, no, from 7 to 10 years, another booster dose from 11 to 18 years, 19 years and older, and we also give vaccines, no, to pregnant patients, particularly at 27 to 36 weeks of gestation. Take note of the timing. So lastly, we have your Legionella, okay? So your Legionella causes pneumonia, and this was first discovered during a convention, no, in Philadelphia. So the air conditioning units actually was contaminated with Legionella pneumophila, and everyone in the legion who attended the convention got infected. So Legionella pneumophila is the model organism causing disease in humans. So it's a fastidious, okay? Fastidious meaning it requires 5 to 10% of carbon dioxide for growth. It's also aerobic, could also grow in aerobic conditions, gram-negative bacteria, and it takes up poorly the gram-staining stains, no? Particularly your safranin. And for it to be stained using Gram's method, you replace safranin with basic fuchsin. It stains better your Legionella pneumophila. However, the most preferred stain for the visualization of your Legionella are actually your silver stains, such as your Warfarin starry and your Litterly stains. So this is best grown in BCYE agar, your buffered charcoal yeast extract. The charcoal here is a detoxifying agent, and the BCYE also contains alpha ketoglutarate, cysteine, and iron, maintained at a pH of 6.9. These are the conditions suitable for a growth of your Legionella pneumophila. So it's kind of difficult to culture this because you actually need, no, you need a certain culture conditions for its isolation. So take note of the agar, no, it's on BCYE agar, it's dark colored because of the buffered charcoal, the charcoal, which acts as a detoxifying agent. And it should maintain this temperature of 35 centigrade and 90% humidity. So it grows slowly such that the colonies only become visible after 3 days of incubation. Take note of that because when you incubate something in BCYE agar and after 24 hours of incubation, you get to see microorganisms or colonies there. It is the Legionella pneumophila. Because remember, you're guided by this principle that Legionella grows slowly and it will actually take at least 3 days for the colonies to be visible. Okay? So take note of that. No? If after 24 hours, 48 hours, you see colonies already in BCY agar, that's not Legionella. Take note because Legionella grows slowly and colonies are only visible after 3 days. So colonies are flat with entire round edges, and the colonies also vary in color. So it become colorless, maybe pink or blue, depending on the strain. And take note, it's biochemically inert. That means it does not produce any reactions to the common biochemical tests that we have in the lab. Legionella pneumophila also causes infection to humans, no? Particularly causing pneumonia. And that is because of these virulence factors. As future doctors, no, you must learn, okay, by heart these factors that are contained, no, that the bacteria has or have, which actually plays a role in disease causation in humans. So we have your a factor causing um, maintenance of its cell wall integrity, thus avoiding killing by the phagocytes. No? Your LCL enhances invasion and cytokine production, triggering inflammation, and then your HSP mediating phagocytosis or antiphagocytic, no, factor, and then your MOMP causing adherence and biofilm formation, your pili, okay, causing helping in the entry of the microorganism, your LPNE, RTXA, and LADC, which mediates attachment and adherence. Then we also have some other factors which is needed for uptake of ferric and ferrous iron. The ferric and the ferrous iron actually needed for its growth. Okay, so we have these virulence factors here and with their involved function, and some other um, factors which also is needed, no, for its isolation or favoring its growth, bacterial growth or isolation. And lastly, it causes, as I've mentioned, your atypical pneumonia, no? Pneumonia, this atypical pneumonia is accompanied by some other symptoms, no? That's why it's atypical. And another condition caused by Legionella pneumophila is your Pontiac fever, which is actually characterized by a flu-like illness. However, Pontiac fever is self-limiting and you actually do not need antibiotics to treat this. And how do we prevent Legionella pneumophila infection is through eliminating aerosols from water sources. No, we kind of do this through putting filters in air conditioning units, no? And then we chlorinate water supply, And we also reduce no cigarette and alcohol consumption, which will actually impair your mucociliary function in the respiratory tract, thus causing infection or making you prone to infection by these organisms. That ends our lecture for today. Thank you very much and God bless.